Good afternoon. I am Barney Graham, Deputy Director of the NIAID Vaccine Research Center on the Bethesda campus of NIH. Thank you to the organizers for the opportunity to participate today. These are my disclosures. I'm an inventor on several vaccines and monoclonal antibodies for viral diseases. We will have an ongoing need for vaccines, both for unmet challenges, including emerging infectious diseases. And while traditional approaches have been important public health tools, they are limited. Fortunately, over the last 20 years, especially over the last 12 years, there have been several new technologies emerge that can be applied to vaccine development, making it more of an engineering exercise. Historically, new vaccines have often been developed because of new technologies. The first two for smallpox and rabies were made by growing attenuated virus in animals before viruses were even discovered. When Goodpasture learned how to grow viruses in eggs, it made the initial yellow fever and influenza vaccines possible. The next cluster came from the discovery of cell culture by Enders and others, allowing the production of large amounts of virus for either live attenuated or inactivated whole virus vaccines. Next was the era of molecular biology, with vaccines based on recombinant proteins, virus-like particles, reassortants, and molecular clones, with production primarily in bioreactors. The recent VSV Ebola vaccine was the first gene-based vaccine vector approved. And I believe the next era of vaccinology will be guided largely by structural biology, with an increasing reliance on manufacturing by chemical synthesis. We thought RSV might be the first example of structure-based vaccine design, but SARS-2 snuck in before and illustrated the power of knowing atomic level structure and the usefulness of many other new technologies. Some of these allow more precision in antigen design and immune response measurements, and others increase the speed of development and manufacturing. This roughly translates into capabilities needed for pandemic preparedness, which is a long-term endeavor, and the tools needed for rapid pandemic response. We have envisioned a systematic approach to pandemic preparedness organized around viral entry mechanisms, working towards a comprehensive knowledge base of structures, reagents, pathogenesis, and animal models by pathogen-focused investigators, and supported by a centralized pathogen-agnostic capacity for immune analysis, process development, and manufacturing. We refer to this as the prototype pathogen approach for pandemic preparedness and response. This concept is derived in part from our work on class 1 fusion proteins that are present on many envelope viruses of concern. In 2013, in collaboration with Jason McClellan, who was a postdoctoral fellow in Peter Kwong's lab, we were able to capture the prefusion structure of the respiratory syncytial virus F protein. This revealed a new neutralization-sensitive epitope at the apex that disappears when F rearranges, as it often does spontaneously. Antibodies to what we call site 0 have 100 to 1,000-fold greater neutralizing potency than antibodies that recognize the post-fusion structure. This explains the finding in at least 5 large efficacy trials done over the last several decades with this molecule that neutralizing activity is only boosted about 2 to 3-fold. However, if you stabilize the prefusion conformation with the C-terminal trimerization domain and internal disulfide and cavity-filling mutations, it becomes a vaccine antigen that can significantly boost neutralizing activity with a single dose and without adjuvant. These types of fusion proteins all work the same way. The protein unfolds, stacking helical domains to project the hydrophobic fusion peptide into the cell. And then the heptad repeat regions pull the membranes together to allow entry of the viral nucleocapsid, and the protein ends up as a stable post-fusion molecule. You want vaccine-induced antibody to target the pre-fusion functional form of the protein, not the non-functional post-fusion form. Class I fusion proteins share functional homology and have similar motifs and domains, although they have different shapes and surface topology. The RSV F protein is very similar to paramyxovirus F proteins, and these virus families have separate attachment and fusion proteins. Other envelope viruses like influenza, HIV, Ebola, and Lassa have a cap over the fusion machinery that serves as the attachment function and also needs to come off to reveal and activate the fusion machinery. Coronavirus spike protein has both the S1 cap and an intervening segment. These shared features can inform not only within virus families, but across virus families. While solving the pre-fusion RSV-F structure, MERS coronavirus emerged in the Middle East. Since there were no available structures for coronavirus spike, and since Jason McClellan was beginning a new faculty position, we decided to work together and apply our RSV findings to coronaviruses. After 3 years and in collaboration with Andrew Ward at Scripps, we obtained a cryo-EM structure for the spike protein of the endemic human beta coronavirus HKU1. A year later, we found that a 2-proline substitution at the top of the central helix not only stabilized the HKU1 spike, but spike proteins from MERS, SARS-1, and several other coronaviruses. Importantly, the 2P mutation not only preserved neutralization-sensitive apical epitopes, but stabilizing the protein significantly increased protein expression from transduced cells, which is an obvious advantage for gene-based antigen delivery. Using the S2P mRNA for MERS in collaboration with Moderna, we showed that human DPP4 transgenic mice could be protected from lethal MERS challenge in collaboration with Ralph Barrick at UNC. We had similar data showing our vaccine designs for the paramyxovirus prototype Nipah could be delivered by mRNA and also protect against lethal challenge. This is where we were when the new outbreak was reported in Wuhan. That is what allowed us to immediately respond by designing reagents and vaccine antigens when sequences were released in mid-January. We worked with Jason McClellan's group at UT Austin, who rapidly solved the structure of the new spike. Once you know that the protein is in the right conformation, it gives you the confidence to develop assays as we did with the CDC, discover monoclonal antibodies as we did with Abcellera and Lilly, and make vaccines as we did with Moderna. Based on the prior work on spike antigen design, several other vaccine developers are also using the 2P version of spike as noted by the red asterisks. Spike or subdomains of spike are the primary vaccine antigens used in the more than 260 vaccine development programs globally. This is another testament to the way new technologies have not only changed the way we think about vaccine development, but have made a variety of vaccine approaches much more accessible to a larger number of investigators. You will hear from others in this session about how several of the vaccines have been evaluated in phase 3 efficacy trials and are now authorized for emergency use and being rolled out to millions of people. Some people have been concerned that the vaccine development process went too fast, but this was the culmination of decades of work, and I believe that if we had been better prepared, we could have saved an additional 2 months. For example, if we had already had phase 1 data on the mRNA vaccine for MERS coronavirus and had done more extensive planning for the clinical development and transition from phase 1 to phase 3, we could have potentially been vaccinating in October instead of December of 2020. The premise of the prototype pathogen approach for pandemic preparedness is that the 120 or so viruses in the 26 virus families known to infect humans would be extensively studied, and prototypes for each virus family or genus of related viruses would have products developed and evaluated through phase 1. The others would be evaluated through animal model testing, and the products would be designed with the possibility of generalizability in mind. This is consistent with the priority list published by WHO and CEPI, but is more systematic and comprehensive and allows for more learning across families. The vaccines for each prototype virus should be fit for purpose, but there are choices that could affect development times. In general, vaccine modalities that use platform manufacturing approaches, especially those made synthetically as opposed to production in cells, will be the fastest. It's possible that protein manufacturing could be extended to a platform approach and achieve shorter development times if producer cells could be generated with more precision. For example, if gene inserts could be put into the same site in the genome for each new product, it may shorten process development times and make it more of a platform approach. The process of identifying generalizable vaccine solutions for the 26 viral families organized by entry mechanisms could begin with the assumptions that each pathogen would have atomic level structure information, that the primary mechanism of protection would be antibody mediated, and that the delivery approach could be either gene-based or protein. For viruses with class 1 fusion proteins, vaccines would focus on both attachment and fusion mechanisms using soluble proteins, membrane-anchored gene-delivered proteins, or nanoparticle display. Most of the viruses with class 2 fusion proteins or non-envelope viruses could likely be addressed using virus-like particles, but other approaches may also be effective. Class 3 and other category of viruses could be addressed with replication-defective viral vectors and other approaches allowing multivalent antigen display. The needed information to advance products is already available for several virus families, highlighted in blue, while others still need considerable work, highlighted in yellow. However, only 4 virus families face what I consider major biological hurdles that still require breakthroughs to have a clear path forward. Chief among these hurdles are extreme antigenic variation or immune avoidance properties that would allow viral persistence. In summary, the COVID-19 vaccine response is a proof of concept for a prototype pathogen approach for pandemic preparedness. Rapid vaccine development was based on prior basic and translational research addressing both precision and speed, and pre-established public-private partnerships that had built trust and allowed rapid decision-making. We believe that the ongoing development and opportunistic use of new technologies, combined with global coordination of research and development, provides a path forward to address future pandemic threats more effectively. And I would like to end by thanking the principal investigators that I work with at the VRC, Jason McClellan, who's been involved in all of our work using structure, and the Viral Pathogenesis Laboratory that I direct, who are a wonderful group of people, and all the collaborators needed to accomplish this last year. Thank you very much, and I'll end here.
It gives me great pleasure to be part of this symposium for the National Academy of Sciences. My job is to give you an update on the status of COVID-19 vaccine development. This slide is one that I made back in April of 2020, shortly after Tony Fauci called me and said that he wanted me to help organize the COVID vaccine clinical trial development program. At that time, I knew that we needed to develop multiple vaccine platforms. No single platform could be large enough to vaccinate the entire world. We needed platforms to cover the field scientifically. Manufacturing scalability was a key factor. We needed to coordinate this effort to involve global vaccine manufacturing companies in a global vaccine effort. And in order to do that, we needed to test manufacture at scale and deliver into people's arms. And this was going to be an unprecedented effort. And all of us have been part of this for the last year, and we know that all of these are true. When we conceptually thought about this, we thought about the fact that we should harmonize the trials. Vaccine delivery was going to be unknown at that time and would likely not be at the same point in time. So give each vaccine an independent evaluation and set up a harmonized system so that we could have some relative comparison between the vaccines, not absolute, but relative. So we did that by harmonizing the endpoints, by having a collaborating clinical trials network, by using common laboratories, to use in our correlates protection analyses, to use a common approach that the statisticians who work in the COVID vaccine program with us would do this, and we do it within and cross protocols. And importantly, we would have a common DSMB, an independent group who would define when the trials reached efficacy endpoints and determine when a trial was futile or non-futile. We've been lucky that all they work, and to make common decisions. So we started this back in April, even before Operation Warp Speed was coined, and we took the HIV networks and the influenza networks, put them into a network that we called the COVID-19 Prevention Network. We grew that network to over 170 sites in the United States, all of academic medicine, the VA, everybody participated in that country in developing the clinical trials program associated with our COVID vaccines. We also had an unprecedented sort of staging of the vaccines, as depending on when they got through phase 1, phase 2 trials. There was no corners cut here. Moderna and Pfizer started in the end of July. AstraZeneca started in the end of August. J&J sort of mid to late September. Novavax in December. And Sanofi won't start till mid-May. Their first batch of phase 1 work trial did not have good immunogenicity, and they started over. Everybody agreed the main goal was to evaluate each candidate vaccine with high veracity for safety and potential efficacy in reducing COVID-19 disease. We designed each trial as 30,000 persons, so almost double the size of what we needed. We wanted 150 disease endpoints. That was the sweet spots for at least greater than 50% efficacy with a lower 30% bound. We knew we needed to, it was critical to enroll the communities affected the most by the vaccine, by the disease. So Black, Latinx, and tribal communities were into each trial. And we wanted to make sure the vaccines, prevented disease, were conducted the trials in the epidemiological setting of persons at greatest risk of the COVID-19's complications, including comorbidities, age, and race. I think we're seeing the fact that both the side effects and the efficacy that we see in these clinical trials appear identical to what the real world data that are coming out of these population-based approaches. Both Pfizer and Moderna were the first vaccines to start, and they were the first vaccines that reached their efficacy endpoint approximately 5 months after initiation of the trial. Sort of a remarkable event that you could enroll 30,000 to 40,000 people and have an answer within a 5-month period of time. The answer was wonderful news. I'm showing this slide. This is actually the Pfizer immunogenicity data, but to show that after one dose, very low neutralizing antibodies, even though there's early effectiveness, antibody titer is low, and you worry about that with respect to emerging variants. Second dose, marked change in antibody titer, way higher than natural immunity, definitely way above background, and good immune responses with the second dose, both with the 65 and younger age range. This is the Kaplan-Meier curve. The blue is the placebo. The red is the vaccine. This is the Pfizer vaccine. You see enormous separation and overall around 93 to 94% efficacy. The Moderna vaccine, identical RNA strand, different soap bubble around it, but the results are the same. 95% overall, 185 endpoints in placebo, 11 in RNA. And whether you were young, old, male, female, comorbidities or not, you actually had the identical effectiveness, high effectiveness, and equal to the Pfizer vaccine. To have two large-scale efficacy trials enrolled completed independently with similar results, I think, is remarkable. The spike is essentially identical, allowing one to feel comfortable about the veracity of the efficacy data. The vaccines were well tolerated. The side effects, second dose worse than the first, but were similar to side effects that we're seeing with the large-scale vaccination programs. I think the similarity of the data means either vaccine can do the job. And it is neat that the real-world data, especially if, let's say, Pfizer here from Israel, where 4 or 7 million people are in this, you see one-dose efficacy of around 43%, 56% after the day 21, and very high efficacy against symptomatic disease, against hospitalization, against medical complications, and now death. At 95 to 100% efficacy of the RNA vaccines in the field. What's the durability? Well, Pfizer just had 6-month durability of 91%. In the United States, it's 93%. And 100% durability against severe disease. Maybe the most interesting thing, that, besides this wonderful data from their 6-month press release, was that they had a small cohort of 800 people in South Africa. They had 9 cases in placebo, 0 in vaccines. 6 out of the 9 were the B1351 variant. So we have some data here that suggests that this Washington Wuhan-based RNA vaccine may have high efficacy in against the South Africa variant, saying that, well, maybe we don't need to have a variant. Maybe either boosting with Wuhan or even the immunity we get with Wuhan over a 6-month period of time, maybe it'll be longer, will have high degree of efficacy against these variants. Third vaccine was the Ad26 vaccine from Johnson & Johnson, one-dose vaccine. This vaccine has much lower neutralizing antibodies than RNA. It has very good binding antibodies. The neutralizing antibodies sort of gradually rise from day 28 to day 72. We see increasing efficacy post day 28. And it has very nice CD8 T-cell responses, better than the RNA. And the data are very good, somewhat different, depending on the variant. In the U.S., we had 72% point prevalence. In Brazil, 68%, slightly less in South Africa, but 87, 88% against severe disease. So slightly less point prevalence against symptomatic disease, but very high prevalence against what's important, severe disease, hospitalization, and essentially 100% more efficacy against mortality. Here's the Kaplan-Meier curves. You can see there's not very good separation, not quite as much. The left side of the curve is moderate to severe disease. The right side of the curve here is severe disease. Very great separation in severe disease and very good efficacy between vaccine on the lower line and placebo on the upper line. In South Africa, this is the Kaplan-Meier curve. You see it doesn't separate till around 28 days. And then you have very nice separation both against severe disease as well as symptomatic disease. This is a Kaplan-Meier curve from the recent U.S. study of AZ that reported the 73% overall efficacy. So very important that we were able to show effectiveness in the U.S. study. 2 doses, 1 month apart. Very nice efficacy against symptomatic disease. Much of that data is starting to come out. One of the issues with the AZ vaccine has been that it did not do well in a trial conducted in South Africa by Shavir Mahdi that's been recently published in the New England Journal. You see that it really had no efficacy, 20 cases in placebo, 19 in vaccines, and about 1,500 people in this South African relatively young cohort, essentially no efficacy. In the same cohort, there were 15 cases early on before the variant was present, and that split was 12 and 3. It's very similar to the 73% efficacy. Why this vaccine has such marked variation in its efficacy between the variant and let's say sort of standard strains is really unclear. It's the only one that doesn't have the proline stabilized spike. Is that related to it? Are there other factors? I think we do not know. The fact that the variant can have reduced effectiveness with some vaccines is present in the Novavax program. The Novavax vaccine in the United States is part of the operations program in the U.S. is ongoing, and that trial is ongoing, fully enrolled. Actually, the double-blind crossover is about ready to occur in April, and we're looking forward to seeing the results whenever the DSMB says it's ready. Novavax did have an independent trial with high efficacy, around 95% in the UK. They did the South African trial here with 4,400. There were 147 cases in the vaccine group and 96 in the placebo group. 5 severe cases were observed in the placebo group. And so here's a vaccine that overall had around 48% in South Africa and 55% overall vaccine efficacy. So in summary, what the scientific community, academia, industry, and government has done in the COVID-19 vaccine development is really remarkable. Everyone, especially we who have worked in HIV, should feel proud about the data that has been generated. Whether it's mRNA, viral vectors, protein vaccines with adjuvants, they're really making their mark and will be the technology that brings us back to social normalcy. They really have high effectiveness. And it's very clear that all of them deliver immune responses or protection that is greater than sort of wild type infection. To date, the data say that higher antibodies are great, but to me, are not the only factor operant in vaccine protection. I think the AD26 data suggests that T-cell immune responses are important, especially for severe disease, that you have sort of differences in the point prevalence, but really similar differences in severe disease. And I think the Novavax data versus the RNA data also say that correlates likely differ by platform, especially for severe disease. And I think there are implications about this with respect to how we look at variants, and do we just use laboratory data to predict what is going to happen. Yes, we've done amazingly well with respect to the understanding the portfolio and the high degree of efficacy and having the availability of this now portfolio of vaccines. We don't have a lot of data on HIV positive persons, especially with variants. We don't have a lot of data on transplant and immune suppressed patients and whether vaccines versus monoclonals are going to be the best approach. And I think that's a very important patient population because of where persistent COVID is and the emergence of variants with persistent COVID. What I think has been relevant for us is the proportion of people who are vaccine or vaccine coverage. It's really not herd immunity. It's really vaccine coverage that we need to think about, because vaccine-induced immunity is greater than immunity from natural infection. I think is more likely to prevent variant infection, more likely to prevent the emergence of variants and reinfection. What criteria should we use to decide if we need strain alteration? I think the recent Pfizer data on the B1351 say that Wuhan-based RNA vaccines may be quite okay against the variant, even though they show 10-fold to 14-fold less neutralizing antibodies. And I'm beginning to feel that good clinical data are needed to make the decision about, well, what we should do with variants. Do we want to always boost with a variant? Will that actually give more original antigenic sin and a narrowing of the immune response over time? Do we need a different kind of priming strategy? Or should we stick with an original prime and have a variant boost when we really need it? I think what is clear is that everybody needs to be vaccinated. Not 20% of a country or 30% of the country, but we, for the scientific community, must advocate to create the infrastructure to vaccinate everyone globally. If RNA vaccines emerge as the one that needs to be the one that has the best longevity, then we actually need to do maybe what we did with antiretrovirals, use tiered pricing and increase the global capacity to make RNA vaccines. That's not to say that the viral vectors vaccines aren't going to make their mark and are making their mark, and maybe the 2-dose studies will even get it up to where the RNA vaccines are. It's important that we have the protein platforms and the viral vector platforms. They clearly are effective. They are very effective vaccines. And we need all the vaccines in order to make everybody on the globe immune from this agent and to decrease the amount of strain variation that we are seeing throughout the world. So it's a privilege to give such a talk. You don't give such a talk without having an enormous number of people helping you and participating with you and counseling you and working with you. To Mike Cohn and Kathy Newsel, who have worked intimately with me in the CoVPN, to our funders, to John Muscola, Barney Graham, and Julie Ledgerwood, who have really been incredible partners, to Tony and Francis, who really showed the leadership that allowed us to even to do this, and to I guess here it's called Operation Warp Speed now, the National COVID Operations Response, to Moncef Slawi, David Kessler, Mary Marich, Merlin Robb, Tina Tong, Julie Aiken, last but not least, Matt Hepburn, who has played an instrumental role on the BARDA OWS side. Thank you.
Thank you for the opportunity to talk to you today about COVID-19 vaccines. Based on years of scientific research and cutting through a lot of red tape, we now quickly have safe and effective COVID-19 vaccines. But of course, our goal is not to have vaccines sitting on a shelf. Our goal is to use these vaccines to control the pandemic in the United States and around the world. And that's what I'm going to talk to you about today, the implementation of the COVID-19 vaccine program. So vaccine implementation has multiple components, and it requires planning and coordination. People have actually been planning for a pandemic for many, many years, but certainly our planning for COVID-19 started in earnest last summer. And here are some of the components of the program. We need to prioritize populations. We need to determine how we're going to allocate vaccine. We need to distribute the vaccine, that is, how to get the vaccine from the manufacturer to the end users. We need to administer the vaccine, that is, the vaccine going into people's arms. And we need to monitor safety, effectiveness, uptake, and also do it all again for those vaccines that require a second dose. This is all underpinned by regulatory considerations for this program, especially because we anticipated that early on there would be limited supplies of vaccine. It was very important to have really real-time data and monitoring systems. And finally, communication and guidance, education and engagement have been and will be hugely important components of this program. The public health impact relies on rapid, efficient, and high uptake of the complete vaccine series, with a particular focus on those at increased risk for severe COVID-19 illness. What we're undertaking as a country is really unprecedented, and it is going to be difficult, but working together, we can accomplish this. As of 3/29, the US has already administered nearly 146 million doses of COVID-19 vaccine. That's almost a third of the US population has received at least one dose. Most of that vaccine has been either Pfizer or Moderna, since the Janssen vaccine was only more recently authorized for use by the FDA. You can see on this map that all places, all states and federal entities and territories have begun vaccinating in earnest. Now, this graphic tries to show the different phases of the vaccine program. And I wanted to make this apparent because we are expecting to be close to a transition point. When we began the vaccine campaign right before Christmas, we knew that the limited supplies of vaccine that were available were going to require us to really have a very focused program. So when you have a setting of constrained supply, you need to have rapid and efficient distribution, but you have to have highly targeted administration to achieve coverage in priority populations. At CDC, we relied on our Advisory Committee of Immunization Practices, but also the National Academy of Science and several other groups that looked at algorithms for how to prioritize the population. And those groups generally all agreed that prioritization should be given to those at highest risk for severe disease and severe outcomes, and also those whose jobs put them in the front lines at risk because of continued possibilities of exposure. These categories were a framework, not meant to be a barrier, and they were implemented slightly differently in different states across the US. As I speak to you now today, most states are either in a 1B or 1C phase. I expect that by the time we get to next month, when I think the rest of this program airs, many states will be moving over to phase 2. Now, phase 2 is what we consider it to be when there are a large number of doses available and vaccination can be really focused on the general public. In this phase, we have sufficient supply to meet demand, and we really have to emphasize rapid, efficient, and equitable distribution. As we get to this second phase, we are really going to need to focus on increasing vaccine confidence. In the first phase, people who are highly motivated are likely getting vaccinated at a faster pace. We need to make sure that as we move forward, no populations are left behind, and we need to focus on engaging and answering the questions broadly across the population, as well as making sure that everybody in the US can get access to the vaccine. And I would say everybody, but I guess for now at least, I mean most adults, since these vaccines are generally licensed in adults. We're certainly hoping that authorized in adults. We're certainly hoping for additional authorization towards adolescents this summer. So to continue this discussion of how distribution and administration is going to be shifting as we move into this farther spring and the early summer. In the early phase of distribution, COVID-19 vaccines are administered in focused areas for priority groups. So if you're vaccinating healthcare workers, long-term care facility residents, and public health clinics, you're trying to reach these focused populations. As we have larger supplies of vaccine available, as we move into the area where we're really trying to move vaccine out quickly to many parts of the population, we need to expand the administration sites. We need to ensure equity and expanding access so that we bring vaccines closer to people to make it easy for them to get vaccinated. And you'll be seeing increasing supplies of vaccines in all of these categories. There are multiple vaccine administration programs that we're relying on to accomplish this goal. Here's just a few of them. We started early with the long-term care facility program to really focus vaccination in long-term care facilities for the residents and the staff, since residents of long-term care facilities have been at such higher risk for disease and for poor outcomes. We've been able to stand up the retail pharmacy program, and in fact, by mid-April, we'll have pharmacies stood up all over the United States. Nearly 90% of Americans live within 5 miles of a pharmacy. The Health Center COVID-19 vaccine program is a program that we're working on with HRSA through FQHCs to ensure underserved communities who are disproportionately affected by COVID-19 are equitably vaccinated. And this program really prioritizes public housing residents, migrant agricultural workers, people with limited English proficiency, and people experiencing homelessness. The community vaccination centers is a collaboration with FEMA, and these are mass mega sites, but as well as mobile sites really stood up in multiple places across the US in collaboration with health departments. We're also really expanding the use of mobile vaccination units with the community vaccination centers and the retail pharmacy programs. And of course, we work closely with our jurisdictions every day who are both coordinating the entire program in their jurisdiction, but also really doing a huge amount of the vaccination in their own communities. Communicating and reinforcing confidence in vaccines is critical in any implementation effort, and definitely true now. I'm happy to be able to say that as we've moved on, increasing number of Americans are saying that they would like to receive a COVID-19 vaccine. In the fall, before there was lots of information known about the vaccines, the numbers were lower. In December, it was 71% of the general public, and the numbers that I've seen with recent few weeks are even higher than that. These factors that weigh on acceptance shouldn't surprise you. People want to know what are the side effects, how well will it work, how safe is it, and how much does it cost? People will believe that COVID-19 vaccines are more acceptable if those that they trust recommend them getting vaccinated, and people still trust their own healthcare team. So the healthcare team saying it's safe will make a big difference. Of course, the vaccines are no cost to the individual. We need to make sure people understand that. People are more likely to be vaccinated if it gets them back to school and work, getting them back to seeing their family and friends. And certainly the ease of getting vaccinated is really important. So we need to continue to reinforce trust. And we also really need to be working to engage communities and individuals, understanding that the trust in your own community and those who give you advice in your community is hugely important to this goal, which is getting everybody access to vaccine, and also making sure that when anybody has access to vaccine, they're ready to roll up their sleeve and get it. So in closing, our goal remains to ensure efficient and equitable access to COVID-19 vaccines for every American. I didn't talk much about it today, but a cornerstone of our program is vaccine safety monitoring. We need to assure the public and ourselves that the vaccines remain very safe, and the data so far is incredibly reassuring. Our new vaccine programs bring vaccine to locations people trust, and as supply increases, we anticipate seeing vaccines being administered in schools, in churches, in community centers, really any place that it makes it easy for people to get access. And finally, confidence in the vaccination program is key. Trust in the vaccine, in the vaccinator, and in the system that produced it are really key to ensuring that people feel good about rolling up their sleeve to get vaccinated. Thank you. Thank you for your attention. Thank you for everything that you're doing. We really all are part of the solution to ending this pandemic. And I think that vaccines will remain an important component to do that.